Yeah, good morning, boys and the girl. After a gap of 1 week, we are going ahead. Just for your memory sake, I am trying to help you with the introduction. We were talking about cell, the unit of life, and we defined cell as the structural and functional unit of life. And we are thankful to these two scientists, Robert Hooke, who discovered the cell, but of course he discovered this in Koch cells, and Anton Leeuwenhoek for observing and describing a living cell. And two scientists, as I was telling you, Schreier and Swan, proposed cell theory. And according to cell theory, the cells have a thin outer layer. We call it cell membrane or plasma membrane. And he also found that plant cells have cell wall. So that is one of the differences between plant cell and animal cell. In plant cell, the outermost layer is cell wall, whereas in animal cell, plasma membrane is the outermost layer. And in animal cell, there won't be cell wall. Externally, it is covered with plasma membrane. And also he said that cells are having inside them what we call cell organelles, like cytoplasm, mitochondria, plastids, ribosomes. All these are cell organelles. And another Russian scientist, he made or proposed the cell theory with one valid point. What is the point that he made? He proposed was that new cells arise from pre-existing cells, omnis cellula, e cellula. That means to form a new cell in any organism, there must be a previously existing cell. So naturally the question comes that where from that initial cell must have arisen. And if you recollect from your memory, I said there is a theory called simple spec theory in the chapter evolution. We'll talk about that again in evolution. That different elements, chemical elements, combine together in a particular ratio in the seawater to form a structure called a simple spec. From that simple spec, the first original living cell must have arisen. So if you analyze, basically we are all made up of what we call basically cells, and cells inside them have cell organelles. I said cytoplasm, ribosomes, Golgi body, endoplasmic reticulum. These are all cell organelles. So starting with, if you analyze, basically we have some chemical elements, chemical compounds. They join together to form cell organelles. Cell organelles join together form cell. Cells join together to form tissues, and tissues together make organ. Organs join together make organ system, like digestive system, respiratory system, circulatory system, like that. Okay? And organ systems together make an organism. So we all covered about these various aspects of the types of cells, and when we say structural and functional unit, when we define cell as the structural and functional unit, we keep in mind that structure is nothing but the shape and size of the cell. So we have classification of cells based on the size of the cell and the shape of the cell. I also said that the size and shape will determine the function. I gave an example. Cells present in the eye are spherical, round, so that we are able to see things, vision, it helps in vision. Cells present in thigh region are cylindrical, so the thigh muscle has to bear the weight of the person. Imagine if it is to be reverse. Cylindrical cells are rectangular cells present in the eye. What would be the shape of the eye? Will the eye having rectangular cells would have helped us to see as we are seeing things today? So it goes without saying that cell structure decides the function and vice versa. Function will decide the structure. That's why we both together we say cell is the fundamental unit of living organism and it denotes structure and function. And based on the nuclear material, whether nucleus is clearly present or not, we divided the cells into two types: prokaryotic cells and eukaryotic cells. Pro means primitive, not very clear. Karyon means nucleus. So prokaryotic cells are cells that have no clear-cut nucleus. No clear-cut nucleus because there is no nuclear membrane. So we will not be able to differentiate which is cytoplasm, which is protoplasm. Remember, we said basically in cell there will be protoplasm. Proto means first formed, plasm means liquid. And once in the protoplasm, nucleus with nuclear membrane is formed, protoplasm is differentiated into two: cytoplasm and nucleoplasm. Cyto means cell, plasm means liquid. So liquid that is present inside the cell, cytoplasm. Nucleoplasm, again plasm means liquid, nucleo means nucleus. Liquid present inside the nucleus. In prokaryotic cell, we will not be able to differentiate, which is the protoplasm, which is the cytoplasm, which is the nucleoplasm. Everything will be protoplasm. So that's how the prokaryotic cells will be, and there will be no membrane bound cell organelles. There will not be any membrane, outer membrane in the cell organelles. So everything looks like a swimming pool. One of the best examples that we can quote under prokaryotic cells is the bacteria. And again, in junior classes you have studied that bacteria are classified into four types based on the shape. That is bacillus, which is rod-like; coccus, round, spherical; vibrio, comma-shaped; and spirillum, spring-like. And you can see all that in the photos. And basically when we see the anatomy of a prokaryotic cell, what is inside, we will see these structures when we take the section of that. Okay, so you have that, you can see the DNA material, but it doesn't have a well-defined nuclear membrane. That's why you said membraneless cell organelles. Same thing, you can see the structure of the outer wall also. Already we have covered. There will be three layers: glycocalyx, cell wall, and plasma membrane. Three layers will be there. And what they are made up of: glycocalyx, cell wall, and cell membrane. The chemical components we talked about. And moving ahead, we said the cell, inside the cell there will be cell organelles. And based on the stain means coloring substance. Okay, thanks to this scientist by name Graham, if bacteria take up the stain and appear colorful, then we say Gram positive. On the other hand, in spite of we putting the stain, coloring substance, if bacteria are not able to absorb the color, then we say Gram negative. Okay, examples also given. And we talk of the next cell organelle, that is Mesozomes. Now I am just expanding that for your memory sake. Look at that, Mesozomes. I hope all of you are able to see. Mesosome, flagellum, cell wall, and look at the nuclear material without any membrane, plasmid and ribosomes. I will be talking about these plasmids separately. Okay, so the mesosomes are the infoldings of plasma membrane, as you have seen. So it includes vesicles, means round bubble-like structures, tubules, small sized tubes, and lamellae, hair-like structures. What is the function of this mesosome? They are for cell wall formation. They help in cell wall formation, and for DNA replication they help, the mesosomes, and for distribution of chromosomes to daughter cells during cell division, and they also help in respiration and secretion. And because of their presence, surface area will be increased so that enzymatic content can be made more. And then comes the next organelle, chromatophore. As the name suggests, chromatophores means coloring substances. So pigments, these are pigment containing membranous infoldings, and the best example is cyanobacteria. Look at that, these are the colorful chromatophores. Okay, and you will be doing an experiment when you come to school, chromatography experiment, okay, wherein you will be able to see what are the various pigments that are present in the leaf of a plant. So we call that experiment as paper chromatography. You will be doing it. And then we said, since the nucleus is not very clear, that's how we are saying prokaryotic, isn't it? And nucleus doesn't have well defined or clear cut nuclear membrane. Nuclear material is spread across, we say nucleoid. We use the word nucleoid. And nucleoid definitely contains nuclear material, including nucleic acids. There are two nucleic acids, as all of you know, DNA, RNA. And in viruses, of course, virus like all the virus that we talk of, right, we say RNA. RNA will be present in viruses. And some of the viruses, of course, will be having DNA also, DNA containing viruses. But most of them will be having RNAs. And then we talk of flagella, that whip-like structure. You can see. Let me expand that. Yes. Are you able to see? Look at that. Flagellum. There are two structures you must note down here. One is the whip-like structure, that is flagellum. Another is hair-like structure, that is cilia. So both will be there, flagellum as well as cilia. And what is the importance of that? This is thin filamentous extension from the cell wall of motile bacteria. That means they help in locomotion, and both for defense as well as offense. Their number and arrangement are varied in different bacteria. So bacterium flagellum has three parts. One is the filament, another is the hook, and third is the basal body. Filament is the thin hair-like structure, okay, it's just like a wire. Hook is the with which it bites, and the basal body is the base with which it is attached to the cell wall. So out of the three, filament is the longest portion and extends from cell surface to outside, as you are able to see. Look at that, right? Next, along with that, there are, I said, small hair-like structures. They are called Fimbriae. Look at that, Fimbriae, these hair-like structures. And pilus, plural, pilae or pilae. So you have structures in the prokaryotic, both pilae and Fimbriae, and these are situated outside. So these are surface structures that help in the motility, and of course they have no role in motility. They are having separate duties. What are those duties, functions? Pilae are elongated tubular structures made of a special protein. That's why the name pilein. The name of the protein is pilein. So in pilae or singular pilus, there is a protein called pilein. And Fimbriae, these are small bristle like, just like in brush, toothbrush you have got bristles, no, like that. So bristle like fibers sprouting out of the cell. In some bacteria they help to attach the rocks. The bacteria will be sitting on the rocks and to the host tissue. It gets attached due to this Fimbriae. That is the function of Fimbriae. Next, we have got very important cell organelles called ribosomes, and many times in your UPSC NDA this question will be there. Ribosomes are responsible for protein synthesis. Remember, protein synthesis. And all of you know proteins are very, very important for our growth and development. Without proteins, there will be stunted growth. The life will be not comfortable. And thanks to these ribosomes, they are present in the cytoplasm and they are spherical in structure, round in structure. So basically, ribosomes are associated with the plasma membrane of prokaryotes. They are very small, 15 nanometer to 20 in nanometer size, and they have got to two subunits. Already they are small, in that there are two subunits. One big bubble and small bubble. That big bubble is 50S and small bubble is 30S. S stands for sedimentation coefficient, which is a measure of density and size. So they together form 70S prokaryotic ribosomes. Here you must not be like in mathematics. So 50 plus 30, 80, but you are saying together 70S. It is, as I said, it is sedimentation effect coefficient. Okay, and as I said just now, ribosomes play a very crucial role in protein synthesis. They are the sites of what we call a process in protein synthesis, translation. Okay, translation. We'll be talking again about this actual process of protein synthesis in class 12th. And several ribosomes are attached to single mRNA to form a chain called polyribosomes. Now, one ribosome is attached to another ribosome. You must have heard in chemistry, your chemistry teacher will be telling you monomer and polymer. Monomer is a small unit. Small units joined together make a big molecule. So that big molecule is polymer. And the small unit is monomer. So here polyribosome, many ribosomes, and the ribosomes of polysome translate mRNA into proteins. Now we will be talking about this at length in class 12th when we take up protein synthesis. At present you just remember that there will be involvement of DNA, RNA and ribosomes in the protein synthesis. Without the involvement of ribosomes, there cannot be protein synthesis. So at present you remember that ribosomes are responsible for protein synthesis. And last we come to that inclusion bodies. That means there are certain cell organelles which are responsible for some of the functions, like these are non-membranous. Remember, that's why prokaryotic. Stored reserve material seen freely in the cytoplasm of prokaryotic. Like, some chemical compounds will be there in the form of inclusion bodies, like phosphate granules, cyanophasin granules, glycogen, and of course vacuoles. And these gas vacuoles found in blue, green and purple and green photosynthetic bacteria. Okay, look at that. This is how. So you have a lot of inclusion bodies, and see the nucleoid because it is not well defined. Ribosomes you can see, inclusion bodies you can see, cell wall, plasma membrane, okay, and of course flagellum and all that we talked about. That is, the other things also will be there. So this is how we talked about the cell organelles, and in this we have only covered the prokaryotic. So normally he will ask you the difference between prokaryotic and eukaryotic. So in prokaryotic, as I said, you just put a table and write on one side prokaryotic, another side eukaryotic. And the first and foremost the thing differentiate you must write is, in prokaryotic the nucleus is not well defined. Under eukaryotic, eu means very clear, karyon means nucleus. Nucleus is very clear. So first difference should be prokaryotic cells will not have well defined nucleus, whereas in eukaryotic the nucleus is well defined. Second, why is it so? Why nucleus is not well defined in prokaryotic? Because lack of nuclear membrane. So second point you must write is, nuclear membrane is absent under prokaryotic, whereas here in eukaryotic you must write nuclear membrane is very much present. Right? Somebody is waiting. Yeah. Okay, Vinod, Arshavardhan. Okay. Yes. Right. So second point that we talk of is the nuclear membrane, presence and absence of nuclear membrane. In prokaryotic there is no nuclear membrane. Because of that, nuclear material spread across the protoplasm. Whereas in eukaryotic, well defined nuclear membrane. So nucleus is clear cut. Having the nuclear membrane, it is very prominent. Third point, in all the cell organelles that we talk of, the mitochondria, ribosomes, okay, including the nucleus, plastids, there won't be any membrane. So we say membraneless cell organelles. Cell organelles will not have any membrane. So overall, the prokaryotic cells will be looking like a swimming pool. Right? There is no clear cut definition. And third, under eukaryotic you can say, eukaryotic cells have got well defined membranous bodies. Every cell organelle will have, mitochondria will have plasma membrane, outer membrane. Plastids will have membrane. So every cell organelle will have a outer membrane. These 3 points you must remember, and of course the other things we talked, that is the inclusion bodies, then presence of flagellum, presence of Fimbriae, okay, presence of pilus or pili, all these. These are the indications of the low character, low evolution. They are just initial stages of evolution. As we move ahead with the evolution, we will be finding that the prokaryotic cells will be evolved into eukaryotic cells. Okay? And particularly when we talk of the protein synthesis, the role of ribosome is enormous. We will be talking about all these things in the future classes. So I hope you have understood all the points under prokaryotic. In my next class, I will be taking up with eukaryotic cells. And particularly in eukaryotic cells, when we take up tomorrow, I want you to practice diagrams. Okay? At home, you practice the diagrams, particularly 2 diagrams. One is the mitochondria and another is the chloroplast. These 2 diagrams you must practice along with the plasma membrane or cell membrane. What is that inside the plasma? So we will be talking not only the morphology of each cell organelle, we will be talking about anatomy also. Anatomy of each cell. What are the chemical compounds that will be present in each cell organelle? And of course, having these chemicals present, what is the function that these cell organelles perform? And when we talk of particularly mitochondria, there will be some tags like the powerhouses of the cell. Most of the time these tags will be asked in your UPSC NDA. Name the powerhouses of the cell. Of course, tomorrow we'll be talking about powerhouse of the cell is mitochondria. We have to justify why mitochondria are the powerhouses of the cell. Similarly, why chloroplasts are the kitchens of the cell, adikimani, okay? And there are some structures like lysosomes. There are some suicidal bags of the cell, we say. Why are they called suicidal bags? Like this, in my next class I will be telling lot of questions, lot of tags that are attached to each cell organelle, and all this will be repeatedly asked in your UPSC NDA exam. So in a way, your preparation for your UPSC NDA has started already, right? With this, I'll be closing today's session. Tomorrow we'll be talking about eukaryotic cells. Yes, any questions on this, boys? Right. If you have no questions, we'll close that, and I'll be giving these notes in your WhatsApp. Right. Take care.